Okay, we're going to talk a little bit about checking fertility in our incubators. There's a couple of different ways to do this. The process, some people candle and look at fertility in the incubators during the incubation process. Others will do the fertility checks at the end during the hatch residue breakout. Today we're going to be initially talking about how we candle. In these types of machines, we'll remove the 6th position and we'll candle this side of the 5th position. Some people might candle at 10 days. Really, the reason why we check fertility after candling is we get enough incubation and enough embryo growth that by putting a light source on the eggs, we can see better what eggs are clear and which eggs have embryos. Again, the eggs that show up clear does not mean they're infertile. We call them unviable. We'll then have to go back later and break those open to determine real fertility, what is infertile and what might be early dead embryos. So we'll start and we'll go ahead and candle some eggs, pull the group off here that are unviable, that look clear, and then we'll move forward to break those out at another time. So the way we do this, we'll start and use a little flashlight, and we'll put the flashlight on the eggs, and we can see the darker eggs containing embryo. When we see a light one like this, we know that is an unviable egg. We collect that, save it, keep going down the row here. Checking for those clear eggs. They kind of glow a little bit like we see here. They glow, and we can then save those to examine at a later point. And this flock is pretty good from what we can see so far. Very few that appear unviable. As we move along, so it makes it a little bit easier to do this in this position here. We don't have to disturb the rest of the incubator by pulling racks out and doing it 10 days. Some people will do a 10-day candling, and that's kind of been a traditional thing. And but this makes it pretty easy to do. We can see here just real quick that we can see this is clear, but we can see a blood ring. So this is an indication, like we said, these are not all infertile. Some are just unviable, so they haven't developed to point of producing an embryo. So this is an interesting egg here. We can see the circulatory system very well. A lot of them are just dark. We see that there's an embryo in here. We can actually see movement of the embryo. At this stage, the embryo, earlier than this, but they're responsive to light. So the light that's in here is causing the embryo to move a little bit. We can see some movement there. So that was kind of interesting to look at. So we would just move along and do all these eggs all on this front row, so we're not disturbing the rest of the egg rack. Okay, so once we get the eggs out of the incubator and we've loaded them on our flat while we're in that incubator, and we will have several of these, we bring them out to a point that a place that we can further examine the eggs. Because again, just because we pulled it out, it looked like a clear egg does not mean it's an infertile egg. We call it an unviable egg. So this is how we would assess and get our true fertility or our real fertility. Again, fertility can be determined on a hatch residue breakout as well. But some people will like to assess that in the machine at a little earlier point in time. So we can take these eggs from the flat, gently take the top off. And again, this is for training purposes. We'll get a little bit slower here. But what we want to do, if we can, is take the shell off so we can see the contents of that yolk and that germinal disc. Often that germinal disc will be raised to the top of the yolk material, so we should be able to see it there. We look for any signs of development. This is where we'll determine if it's fertile or infertile. An infertile egg is then going to have a very tight, compact, white daughter mass around that germinal disc area. If fertility occurred and there's any development from that, we start to see a donut-shaped ring over that germinal disc area, because that's where the embryo will continue to grow. So we look at the egg, make a determination. That is an infertile egg. Again, most of these will probably be infertile eggs, unless we've got some other issues. Again, so this is another infertile egg. Often at this point in time, we see the yolk has maintained the same color, same structure. The membrane, we're breaking at the air cell end. The membrane is clear like it would be on a fresh egg. Often we'll see on eggs that have embryo development, we'll start to see a opaqueness of that membrane starts to turn white to various stages. So this looks like that is an infertile egg. The yolk looks normal, everything looks normal. And this one, immediately, as soon as I start opening and I see the membrane is very white, as we pulled this, we can actually see the embryo here. Small little remnants of the embryo. If you look real close, we can see right here would be the embryo itself with the blood support that was around it. This blood was wrapped around that yolk, and as we pulled it apart, it came apart a little bit. So now we know this was a fertile egg, and we had some early dead mortality in the first 3 days from that egg. There's another early dead. You can see the look of that membrane under that air cell, the yolk, it all has a different appearance and texture. We can see the embryo right here. And if we want to pull that embryo out and really examine it a little bit more, we can see it right here. When I do my residue breakout, one of the things I look for is the pigmentation of the eye that occurs in the 4th day. So 4 days of embryo development, you'll notice eye pigmentation. We can see the eye pigment right here on the end of this embryo. So that would indicate that made it to 4 days. So when I record these, it'll be 4 to 7 day embryo. Obviously died before 8 days when it would get the egg too. So that's a very good example there of a 4 to 5 day embryo with that pigmentation of the eye. So this is an infertile egg. You can see the germinal disc, may not be able to see it right there, where we can see there's no development, no ring, just a very tight, compact little dot. And we'll have some other pictures of this we can show at a later time in this training video, and we can see that germinal disc right there. So this would be the process of, once we determine the unviable eggs out of the incubator, we then need to distinguish between fertility and infertility, because those two issues need to be addressed differently. We can't address fertility the same as we would early dead. So calling all unviables infertile is just not going to work in assessing our hatchery and determining where we need to go to troubleshoot problems. Okay, so now that we have candled the eggs, we've determined the eggs that are unviable. We don't know if they're infertile or not at this point. They're just unviable, meaning we don't see a growing, developing embryo in that egg. So then we take the eggs out, and they need to be broken open to be further examined a little bit more closely. We showed you how to do that. So now let's go through a few other things that have to do with determining fertility and infertility in our broken out eggs. First, the fertilization process, we have insemination of the hen, whether it be natural or artificial. The hen stores sperm. Sperm has to be transported up the oviduct to the site of fertilization before we can actually have successful fertilization in the hen. The hen, when she lays an egg, within 20 minutes she will ovulate the egg that will be laid the next day. When that ovulation occurs, fertilization will occur within minutes after that, before it gets into the oviduct. So right away we have a fertilized egg at the upper portions of the oviduct. That egg then is transported down the oviduct through the portions of the oviduct that are responsible for albumin, thick albumin, thin albumin production, shell membrane, shell. All these things need to occur in a 24 to 26 hour window. The body temperature of the hen is 40 to 41 degrees Celsius, or about 104 to 105 Fahrenheit, which is obviously warm enough for embryo growth. So when that egg is laid, we have anywhere from 40 to 60, possibly more, thousand cells of that embryo, and this is why we can often determine the fertility of an unincubated egg. That fertility will occur at that germinal disc area. On that yolk, the white portion, that's where the female genetic material or pronuclei resides, and so sperm must be attracted to that site and fertilize the egg. So we see an infertile egg on the left here, very tight, compact white dot. On the egg on the right, we can see is kind of a donut-shaped ring around that germinal disc. That represents the cellular development that's occurred during formation of that egg. Of course, when that egg is exposed to proper temperatures to further develop, we start seeing that donut-shaped area get a little bit bigger. It
continues to grow a little bit, even a little bit bigger at 24 hours. So this is the thing, what we're looking for. We're looking for that donut-shaped area that represents the cellular development. Not meat spots, not blood spots. Now this strictly just cellular development. Now, as we go on to a residue breakout, it's a little bit more difficult to see this, but we're still looking for the same pattern of cellular development in that donut-shaped fashion. So once we do this, we can definitively determine fertile and infertile egg, and that is very important in our system to be able to make that distinction.